The nitrogen cycle is one of the most commonly mentioned processes in the fish keeping hobby, but it's rarely explained in any real detail. You'll hear it referenced constantly, usually as a kind of a background process that just needs to be there without much attention given to what is actually happening inside your enclosed system. But once you start looking closer, it becomes clear that this is not a single event or a simple concept, it's a continuous sequence of biological and chemical transformations, happening across surfaces driven by microorganisms and constantly adjusting to how much waste is being produced at any given time. So in this episode of The Science Behind, we're going to break that process down step by step. We'll start with why the nitrogen cycle exists in the first place, and then move into how waste enters the system, how it's transformed through different stages, and what ultimately happens to it once it reaches its final form. Because once you understand each step in this sequence, the nitrogen cycle stops being this abstract idea and starts becoming something you can actually follow as it happens inside the tank. Welcome to The Science Behind. Before we get into what happens inside a tank, it helps to step back and look at where the process comes from, because the nitrogen cycle didn't start with aquariums. It's something life had to figure out long before any of this existed. Nitrogen sits at the center of biology. It's part of amino acids which build proteins, and it's built directly into DNA. Every living organism depends on it in some form, but the way nitrogen moves through a system isn't straightforward, and some of its forms create problems very quickly. Ammonia is one of them. It's produced whenever organic material is broken down, whether that's waste from animals, decomposing food, or cellular processes. In water, it dissolves immediately and becomes part of the surrounding environment. The issue is that ammonia is highly reactive, and in aquatic systems, even small amounts can interfere with basic biological functions, and especially in organisms like fish, where it can affect respiration directly at the gill surface. So once nitrogen starts moving through a system, it doesn't actually take long before ammonia begins to build up. And if nothing interacts with it, it accumulates. Early in Earth's history, this was a constant pressure. Organic matter was being produced and broken down, releasing nitrogen in forms that could not just sit there indefinitely. Something had to process it, or environments would become increasingly hostile and toxic to life. This is where microbial metabolism enters the picture. Long before plants and animals, microbial life was already adapting to whatever chemical energy sources were available. Oxygen was not consistently present, and light was not always accessible, so many of these organisms relied on inorganic compounds to sustain themselves. Among those compounds were nitrogen-based ones. Certain bacteria developed the ability to oxidize ammonia, using that reaction as an energy source. And in doing so, they convert ammonia into nitrite, and other groups follow by converting nitrite into nitrate. These organisms are known as chemolithoautotrophs, meaning they derive energy from inorganic chemical reactions rather than from organic material. As these processes established themselves, nitrogen stopped accumulating in a single form and instead began moving through a sequence of transformations. Each step reduced the immediate impact of ammonia while keeping nitrogen within the system in a different state. That sequence is what we now refer to as the nitrogen cycle. It exists as a result of microbial activity interacting with the chemistry of the environment, gradually shaping a system where nitrogen can move without continuously building up to harmful levels. So when we talk about the nitrogen cycle in an aquarium, we're then looking at a small, contained version of something that has been happening at a global scale for a very long time. And that's the idea to carry forward. The nitrogen cycle is simply how life learned to deal with nitrogen moving through a system. But in aquatic systems, we are taking that and moving it into a very confined space, and that's where issues can come. Once the system is running, nitrogen is already moving through it, and it starts with ammonia entering the water continuously as a direct result of normal biological activity inside the tank. The main source is the fish themselves. As fish process protein, nitrogen is released as ammonia and diffuses out through the gills straight into the surrounding water. It doesn't collect somewhere first, and it doesn't wait to be broken down. It is introduced directly into the system in dissolved form, which means that it spreads immediately and becomes part of the water that the fish are constantly passing over their gills. At the same time, you have additional input from the rest of the system. Uneaten food will begin to break down. Organic material will accumulate and decompose, and microbial activity converts that material into ammonia as well. This happens over a slightly longer timescale, but it feeds into the same pool, increasing the overall load that the system has to deal with. So ammonia is always being added from multiple directions, and it doesn't remain unchanged once it's in the water. It exists as two forms in equilibrium, NH3 and NH4+. And the balance between them shifts depending on the conditions in the tank. At lower pH, more of it is present as ammonium, while increasing pH shifts that balance towards free ammonia. Temperature plays into this as well, with higher temperatures pushing equilibrium further in the same direction. And that shift matters because the two forms behave very differently at the biological level. Free ammonia can move across gills membranes more easily, which means that it interacts directly with the tissues responsible for gas exchange. Even at relatively low concentrations, it starts to interfere with that process, and because it is dissolved and evenly distributed, exposure is constant rather than occasional. So from the moment ammonia enters the system, it is already part of the environment the fish depend on, and its impact is tied not just to how much is present, but to the conditions that determines its form. And since it's being produced continuously, the system has to keep up with it just as continuously. Once ammonia is in the water, it becomes the starting point for the first biological step in the cycle, and that step depends entirely on a specific group of microorganisms that have to be physically present in the tank. These are ammonia oxidizers, often associated with groups like nitrosomonas, and they doesn't exist evenly throughout the water. They live on surfaces. So filter media, substrate, glass, plant leaves, anything that really stays stable long enough becomes a place where these organisms can attach and grow. So instead of thinking of ammonia being processed in the water, it's actually more accurate to think of water carrying ammonia past these surfaces where the actual work is being done. As that water moves across the surface, ammonia comes into contact with these bacterial layers, and that's where the conversion starts. The bacteria use ammonia as part of their energy process, converting it into nitrite in the presence of oxygen. And that detail actually matters a lot. This step only happens where two things meet at the same time, ammonia and oxygen. If either one is limited, the process will slow down, and that's why areas with poor flow or low oxygen don't really contribute much, even if ammonia is present there. Now, even though this reaction is happening continuously, the system handling it has its limits. These bacteria grow slowly, and not just slower than algae or plants, but slow enough that any increase in ammonia takes time to be matched. When more ammonia enters the system, the existing bacterial population can only process what it's capable at in that moment. Expanding that capacity takes time because it means growing more of these organisms across available surfaces, and that growth depends on space. If you have more surface area, you have more room for these bacteria to establish, which increases how much ammonia can be processed at once. If surface area is limited, then capacity will also be limited, regardless of how much ammonia is present. At the same time, this layer is not fixed. Changes in oxygen, temperature, or general stability affects how efficiently these bacteria operate. Disturbing surfaces, cleaning filter media too aggressively, or disrupting the biofilms reduce the active area where the process takes place, which will then lower the system's ability to keep up. So what you need to end up with is a system where ammonia is constantly being added, and a biological layer that has to continuously match that input using oxygen, surface area, and time to do it. And when those two don't line up, when ammonia is entering faster than it can be processed, this is where the first imbalance will start to show. Once ammonia has been converted, it doesn't mean that the system is safe yet. What you're left with at this stage is nitrite, and that's where the second part of the process begins. Just like before, this step depends on another group of organisms that live on surfaces throughout the tank. These are nitrite oxidizers, commonly associated with groups like Nitrobacter, and their role is to take nitrite and convert that into nitrate. So again, it's not happening in the water itself, because water is carrying nitrite across surfaces, and wherever these organisms are established, the conversion takes place at that specific point. At this point, it helps to understand why this step matters as much as it does. Nitrite interacts directly with how fish handle oxygen. Once it enters the bloodstream of the fish, it affects the ability of blood to carry oxygen efficiently, so even if the water itself is well oxygenated, the fish are not able to use that oxygen properly, and that's why exposure can show up as stress, rapid breathing, or lethargy. even when everything else in the tank appears normal. And just like with ammonia, this is not about large spikes only. Even in relatively low levels, if sustained, begin to interfere with normal function. So the system now has to take nitrite and move it one step further. These bacteria oxidize nitrite into nitrate, again using oxygen in the process. The same limitations apply here as in the previous step. The reaction depends on oxygen availability, it depends on water moving across active surfaces, and it also depends on how much of this bacterial community is actually present, because they also grow slowly. So if nitrite begins to accumulate, it usually means that this part of the system has not yet reached the capacity needed to keep up with what's coming from the previous step. There's a lag here as well, where ammonia may already be getting processed efficiently, but nitrite starts to build because the second stage hasn't caught up. Once nitrite is converted, what you're left with is nitrate. And nitrate, although it sounds similar, behaves very differently in the system. It's far more stable and it doesn't interact with fish physiology in the same immediate way, and it tends to accumulate rather than cause rapid acute issues. So that's also why tanks can run for long periods of time with rising nitrate levels without any obvious signs of stress in the livestock in the short term. So this stage acts as a transition point. You're moving from a form of nitrogen that directly interferes with biological function into one that remains in the system in a more stable form, building up over time instead of causing an immediate disruption. And because nitrite sits right in between those two states, it often gets overlooked, even though it represents one of the more sensitive points on the entire process. Once nitrite has been converted, what you're left with is nitrate, and this is where the process starts to slow down in a different way. Unlike ammonia and nitrite, nitrate doesn't immediately interfere with basic biological functions in the same direct sense. Fish can tolerate it at much higher concentrations, and because of that it tends to stay in the system rather than causing an immediate response. So instead of being processed quickly, it accumulates. Every time ammonia enters the system and moves through the previous steps, it eventually ends up here, and unless something removes it, the total concentration will gradually increase over time. There are a few ways to get this handled. Plants take up nitrate as a nutrient source, incorporating it into their own growth. In a system with enough plant mass and active growth, this can make a noticeable difference because nitrate is being continuously pulled out of the water and turned into biomass. But that uptake depends on light, CO2, and the overall plant health, so it's not constant and it rarely matches the total amount being produced on its own. Then you have water changes. This is the most direct way to reduce nitrate because it physically removes it from the water column and replaces it with water that contains less. It doesn't process nitrate, it resets the concentration, which is why it remains the most reliable method in most setups. There's also another pathway, but it requires a very specific set of conditions. In areas where oxygen is limited, certain bacteria can use nitrate as a part of their metabolism, reducing it further into nitrogen gas, which then leaves the system. This process, known as denitrification, only takes place in low oxygen environments, typically deep within the substrate layer, or with specialized filtration where the water movement is restricted. In most standard aquariums, those conditions are limited or inconsistent, which means that this pathway doesn't really contribute enough to fully balance what's being produced. So what you end up with is a system where nitrogen enters as ammonia, moves through a series of transformations, and then largely remains in the form of nitrate unless it is actively being removed. And that's also where the idea of a cycle can actually become slightly misleading in practice. In a natural environment, nitrogen can move all the way up through and return to the atmosphere, completing the loop. In a typical aquarium, that loop is only partially in place, and the final step is usually handled externally through maintenance rather than internal biological processes. So while the system continuously processes ammonia and nitrite, it doesn't fully close the loop on its own, although plants can do much. Everything in your tank is constantly feeding into this process. Fish are releasing ammonia straight into the water, organic material is breaking down, and all of it is being picked up and transformed step by step across surfaces you don't really see or even think about. Filter media, substrate, glass, plant leaves, that's where the system actually lives, and it's not static. The amount of ammonia going in changes The capacity of the system will shift over time, conditions will move slightly, and the entire process will then adjust with it. Sometimes it will keep up, sometimes it lags behind, or sometimes it can crash. And that difference is what determines whether the tank stays stable or will start to drift or inevitably crash. Once you start looking at it in that way, the nitrogen cycle stops being this vague concept in the background and it becomes something you can follow, something you can influence, and something you can build around. And that's really the shift. Thank you for watching this episode of The Science Behind. As always, I'm on the lookout for more topics to dive into, so leave your suggestions in the comments. I try my best to read and respond to everyone. If you enjoyed this video, you might also like another one from the series. There's a playlist of all the Science Behind videos on the screen. Or you may be into aquascaping, I also do some of that. Anyways, please do consider subscribing. Thanks again for watching.